Okay, so this is lecture 23. Just a note, a reminder, we won't be having an in-person lecture on Friday. You can use that time or any time really to watch the lecture that's been pre-recorded by Kim. And this is about how to write your scientific report. So it's really important that you watch this so that you know how to write it correctly and you can maximize your grade. So again, this room will be available if you don't have a place to sit during the day and access internet, so you can come in here and watch it if you want. Otherwise, you can watch from home. So we've been looking at action potentials, and it sort of all starts out with the resting potential of a membrane. And so the resting potential of the membrane is based on the concentration of these ions. All right, so we have sodium, which is at a higher concentration outside the cell than in. We have potassium, which is at a higher concentration inside the cell than out. And importantly, these are both univalent, positively charged ions, meaning they have a single positive charge. And then we have chloride, which is higher, again, out than in. And this carries a single negative charge. And this is looking at a couple of different cells. We have the squid axon, which we talked about. This was used, I think it might still be used, but it was used in a lot of early studies because you can actually physically see this. It's thick enough that you can visualize it. You can take it out of a squid, put it into a petri dish or whatever you want to work with it in, and you can see it. And then the other one we have up here is a cat neuron. So when we're looking at here are these electrochemical gradients, which make things a little bit more complicated than the concentration gradients we were talking about earlier. Because as we can see here, the positive charge of the potassium is balanced by negative charge inside the cell. And that negative charge comes from a variety of molecules, including chloride ions, but also things like RNA and protein carry a large negative charge. So what that means is that when a positive ion steps out of the cell, that's good from the chemical ratio point of view, but you're leaving behind charge. And so they need to balance out this electrochemical gradient. And we can calculate this utilizing what's called the Nernst equation for a single ion. Okay? So the cell is working hard to maintain these concentration gradients. We can plug those values into this equation, and this tells us what the membrane potential will be. And just as a note, when we calculated this for potassium, the membrane potential was, I think, around minus 90 millivolts, whereas if we were to calculate it based purely on sodium, it was about, I think, plus 65 millivolts. So this is the overall problem. We have these three competing ions that all contribute to the membrane potential. So what is the resting membrane potential? Because we have sodium, potassium, and chloride. And the way that we can, and I should point out that these have varying permeability through the membrane based on these leak channels. So the leak channels are important because and these, remember, it's a very slow stream of ions compared to when we open an ion channel. All right, so a little bit is moving through, but it's important because what that means, for example, if the potassium falls too low in the cytosol of the cell, that's going to affect the membrane potential. So the way this works is that potassium can sort of generally be leaking in or out, and we can then utilize the pumps, the active transport, to maintain those specific concentrations. So overall, to calculate the membrane potential, we can use the Goldman equation. And what that combines is not only the concentrations in and out. Okay, sorry, we lost the... So not only the concentrations in and out of all three ions, but also the relative permeability. And so if you punch in the numbers here, this for squid where the relative permeability of sodium is 4% of potassium, so that means we multiply it by 0.04, that's 4%. Chloride is 45%, so we multiply by 0.45. For this, we end up for squid with a resting potential of about 0.0625, which is about minus 62.5 millivolts approximately, which is kind of somewhere between what we calculated for sodium and potassium, which maybe isn't surprising. So we had kind of finished off here looking at what happens during an action potential. And so it's initiated by opening a sodium channel. And so that's happening here at time 0. I should say preceding that, we're at the resting membrane potential, about minus 60 millivolts. 0 is showing, and this is in milliseconds when this whole thing starts, and it starts with sodium channels opening. And that allows sodium to flood in. So if we look at the Nernst equation, remember we can utilize that when the permeability of one ion is much higher than the others. Because what happens is the permeability, say we open the sodium channel and it's very high, the permeability, the relative permeability of potassium and chloride at that time is about 0. And if you negate those, cross those out, you end up with the Nernst equation. So what we start doing is it starts to look more like, it starts to become a more positive membrane potential because we know that's what sodium prefers. And so if we look here, we get this what's called depolarization, right? So we stimulate, we start to depolarize, and we hit some maximal point at which the sodium channel closes. So these channels don't stay open for very long. At that point, we open the potassium channel, and so now potassium floods out of the cell. Okay, so it moves down a concentration gradient, both chemical and electrical concentration gradient in this case, and we get repolarization, and sometimes we go so far as to get hyperpolarization, because we remember the resting potential, if we were just looking at sodium alone, would be about minus 90 or so millivolts. And so the next step is that we flip the sodium and potassium around, because what's happened is at the end of this potential, we have higher than normal sodium inside the cell and higher than normal potassium, well, lower than normal potassium inside the cell. So we use that exchanger to reset that. And at that point now, the refractory period is over. The refractory period is over, and another action potential can travel. Okay, so we have to reset the ion concentrations before another action potential can move past this point. And if we look at the time, this is all occurring in less than a second. It looks like about 5 milliseconds. Okay? And so that was pretty much where we got to before the fire alarm went off. Such excitement. It turned out it wasn't a fire, so everything was okay. Yeah, they seem to be doing a lot of these drills without letting anyone know this year, which is odd. So today we're going to look at the rest of synaptic signaling. We're going to concentrate a little bit on the channels. And then we're going to move on to our next section, which is cell signaling that's not synaptic signaling. So just to hold the date kind of thing, because I know this is our last class this week, and next week we have our, is reading break. Most people know that. So we don't have classes Wednesday, Thursday, Friday next week, right? But we do have classes Monday and Tuesday. Keep in mind, classes are recorded, so I understand if you need a break that you might not want to come into class next Tuesday, so they will be recorded. And I'll have my usual office hours on Monday, so from 1 to 3. And At that point, you can come in and review your midterms. So I'll put increased feedback on today so you can see which questions you got wrong, and then you can talk to me about that next week. And if you can't come during office hours, send me an email and we'll try to arrange a different time. So the week after that, we have our next social science talk, I guess, or social hour. And so for this one, it's pretty cool. We have two people. We have Alyssa Herkel, who was she was in the very first class I ever taught at UVic, which was a third-year cell bio class. And she's an embryologist working for Pacific Center for Reproductive Medicine. And we have Dr. James Graham, who hasn't sent me a picture yet, but anyway, he's an OB-GYN who specializes in fertility treatments. So, and he's actually someone I've known for, I think, 40 years. So anyway, so they're going to be our guest lecturers on November 17th. It should be really interesting. You'll get, there's lots of science, but also ethical concerns when we're talking about fertility treatments. So I think it's something you might find really interesting. And Alyssa can tell you how important my cell biology classes are to succeeding in the world. So, yes?
Is Alyssa a current med student?
No, she's an embryologist. So, yeah, it's a different, she'll tell you all about her path to becoming an embryologist, but it's not an MD position. So, yeah. And then for James, he's a classic MD who took a specialization. Yeah. Okay, so that's November 17th at 6 p.m. It will be online, although actually the three of us we're trying to arrange it so that the three of us will be in the same room. James is one of my bubble people, so I don't know if people still have bubbles, but I do because I worry so much about COVID. So, all right. So how do they're called voltage-gated channels, the sodium and the potassium. And I know more about the potassium ones. We teach those in third-year biochemistry. And there seems to be it's a little bit clearer what's happening in them. But the functionality is basically the same for both. So the way it works is that you have this channel, and it can be open or closed. And both of them, whether it's the sodium or the potassium, are specific to that ion. Again, a very small amount of other ions may move through there, but we can basically call them specific. It's a very small amount that goes through that's non-specific. And it's regulated when these open and close, and they're called voltage-gated because they're regulated by the membrane charge. And so in these examples, what we have is this is the extracellular space or extracellular matrix. And at the bottom, we have the cytosol. And we know that initially the membrane potential is negative, right? Around minus 60 millivolts. So that's the resting state. As a component of the potassium channel, it has an amphipathic helix, and this helix, one of the sides of it is positively charged. So remember, we can think of a helix kind of like a cylinder. So think of all of the residues on one side of that cylinder. So here's a cylinder. Say all the residues facing you are positively charged. So things like lysines and arginines. So those are going to be attracted to the cytosol, right, through electrostatic interaction. And what that does is as that helix comes down, it squeezes the channel closed. What happens when we depolarize the membrane, remember, it becomes positively charged. You now have a charge-charge repulsion. And so the positive charge on the helix is pushed away and up, and that relieves the constriction of the channel and then allows the ions to flow through. Sodium and potassium channels have these specificity gates that will allow either sodium or potassium through, but not other ions. The mechanism of that we'll look at in third-year courses. So that then is what causes these channels to get pushed open. All right? They don't stay open for long because they have what's called an inactivation domain. So this is all part of the same protein, and it's kind of tethered to it. And the best way to think of it really is if you have one of those super annoying oldie-timey bathtubs or sinks that has the little plug on a chain. Does anybody have these? And you're trying to like just let water run and the stupid thing keeps falling in the plug. That's basically what happens here. You have this domain, which actually it's partially attracted into the channel as well. But what happens is it's held in place and it's attracted to the channel. So when the channel opens, they kind of circle and very quickly shut that channel down. And if they don't shut that down, it causes certain neurological deficiencies. Yes?
And what is that called again?
An inactivation domain for that, yeah. So they require this inactivation domain to very quickly shut themselves off. Yeah. This is the figure from a different textbook. This is the one from your textbook, and actually kind of prefer the simplicity of this one a little bit, but you can use either one to study those. The absence of that domain in Drosophila. So Drosophila are interesting organisms. They're a model for studying genetics of multicellular eukaryotic organisms. And their genetics are very well defined and have been used for a lot of study. And some of you may know this, but they often give them kind of interesting names. So sort of funny names. There's one called Techno Trousers and Cheap Date. There's a whole bunch of these weird names. But the one for this is called Shaker. If you inhibit that or you get rid of that domain, what happens is when you anesthetize the flies, they start to shake around. They start to move, sort of jiggle. And it's because their synapse, or sorry, their axons, their neuronal signaling is totally messed up by this mutation. So it's an important feature of the ion-gated channels that they shut off very quickly. So the action potential then is this thing that's going to happen. We're basically looking at sort of one bit of it, but we haven't discussed how this actually drives down the nerve. And so the way it works is that it's initiated here at the axon hillock, and it moves through as a wave. So if we imagine the sodium channel's open, we get a positive charge, and then the potassium open and it closes. So that's what we've been looking at. But what happens is that positive charge opens the next sodium channels. Okay? So I'm a sodium channel, potassium channel. Sodium opens, positive charge comes in, and then the potassium opens to relieve it. But by then, some of that positive charge has moved down here and open the next sodium channel, which will then quickly inactivate. But by then, and so this opening and closes pushes the signal down the axon towards the synapse. And that's described here. So it's this wave of depolarization, repolarization that's physically pushed forward because the sodium channels open ahead when they get some positive charge, and they will further open, and then the potassium open to shut it off. So that drives this whole thing forward. There is more to this, though, because this system as it's drawn would actually not be efficient. The axons themselves are what we call myelinated, okay? And this myelination is the product of two types of cells, the Schwann cells and the oligodendrocytes, which I think I have misspelled on your slides as oligodendrites. So you might want to make sure they're called, and there it should be oligo, not oligio. should be oligodendrocytes. And for these, we have Schwann cells in the periphery nervous and oligodendrocytes in the central nervous system. And these act as an insulator, and they physically wrap their membranes, their plasma membranes physically wrap around the axon. And the lipids in that membrane are highly enriched in galactocerebroside, so that was a glycolipid, single myelin, and cholesterol. And there's other protein components as well. So this slide shows us what it looks like. So here we can see the Schwann cells themselves. The actual cell wraps itself around the axon. Whereas for the oligodendrocytes, they have these sort of processes, these parts of the membrane are pulled out and then wrapped around. Okay? Yes?
So just to clarify, a Schwann cell will wrap a single axon, whereas an oligodendrocyte can wrap multiple.
I would think of it more as that a Schwann cell wraps, the actual cell itself wraps around. And the oligodendrocyte sends out these, which is, I guess, the name, the dendrites, that go out. They could potentially wrap around multiple, I guess, but yeah, it's just sort of a difference in how that goes. So what this does now is this alters the landscape a bit in that instead of moving, as we saw before, just to the one next door, which is going to be quite slow, what this has is there's gaps in the myelination, and those gaps are the only places where we have the channel proteins that can open and close and let ions through. So the spaces where it's myelinated, no activity occurs. So what this means now is it jumps. It jumps from these one gap to another. And these gaps are called the nodes of Ranvier. And I think that's labeled at the top here. These nodes of Ranvier. And so what happens is these leap forward now. And these nodes can be 1 to 2 millimeters apart. So on a cellular scale, that's a big distance, right? 1 to 2 millimeters. If we think like an average animal cell is like 50 micrometers, these things, we know the axons are very long, and these can be spaced that much. And so it's much more efficient for them to function this way, and it rapidly speeds up the rate at which the oxygen potential can move down the axon. And when we lose myelination, that can result in neurological disorders, and the one that comes to mind is multiple sclerosis. What happens is the myelination of the axons is lost. Okay? Any questions about that? Yes?
So how does, I'm assuming ions are what's moving through the nodes of Ranvier?
Oh, so at the nodes of Ranvier, that's where the actual channels sit.
Okay.
So the channels sit there, and that allows the membrane that's where the ions move through, but it allows it, instead of moving just a little bit each time, it allows it to leap from one to the next. So it just improves the efficiency. Yeah. Okay, and so finally, we arrive at the synapse and the synaptic bouton, and what happens here is the positive charge of the sodium, it opens another channel, and this is a calcium channel. So that positive charge opens a voltage-gated calcium channel. We remember that calcium is at low concentration in the cytosol. So what happens is when this opens, calcium floods in, and this is what causes the fusion of the neurotransmitter vesicles with the membrane at the synapse and the release of the contents. And so that, of course, is mediated by the snare complexes, the syntaxin, synaptobrevin, and the SNAP25 that we looked at earlier. And again, there's a video now posted on Brightspace that shows that fusion of the vesicles containing neurotransmitter and the role of the snares. And so that's the end of our section on synaptic signaling. So a cool story that ties together sort of three concepts we've looked at: the importance of ion gradients and membrane impermeability, channels and membrane transport, as well as our vesicular transport. So those are all tied together here. Now we're going to look more at other types of cell signaling. Okay? And so when we look at cell signaling, pretty much all cells do some form of signaling. Some is pretty rudimentary, so kind of basic, and some is really complex. If we look at organisms themselves, and you can see here, multicellular organisms appeared like maybe around 2.5 billion years ago. Sorry, unicellular organisms existed 2.5 billion years before multicellular appeared. And so a unicellular organism can have quite a lot of complexity in its signaling, but it's much lower than the signaling complexity we see for multicellular. So with a multicellular organism, it doesn't just interact with the environment and then with kind of like neighboring cells. It's integral. It's part of an entire organism. And so the signaling pathways can be really complex. The signal molecules, they're generally at pretty low concentrations. And so you're not like bathing yourself in masses of these. And the receptors generally have a strong affinity. So affinity is how well something binds. So they're really good, the receptors are really good at binding to the signal. In this case, we'll also sometimes call the signal molecule, we're also going to be calling that ligand. Okay, so the signal or the ligand is what binds receptor. And so the bottom part here is really important. And I also want to point out something that sort of arises from this. When I teach you a signaling pathway, or in third year when you learn a signaling pathway, and even to some extent in fourth year, when we draw you a signaling pathway, we show you it as a pathway. A activates B, activates C, activates D, and we give you this more or less linear path. There's a huge number of these linear paths, however, and they're integrated. So they crosstalk constantly with each other. And I will be showing you slides for this, but I want you to always keep in mind that these pathways are not occurring in isolation, even though as I teach them to you, we're going to be teaching them to you in isolation. And so the actual output from a cell is a result of all of these signaling pathways at any given time. Right? So all of those signals are brought in, processed, and that leads to whatever our cellular output may be. Here's a few of the types of signaling we can look at. So in this particular example, so here's the cell is on the left and the output's on the right. And these signals shown in blue, A, B, and C, these are survival signals. So cells have to be constantly told to stay alive. And if you stop telling a cell to stay alive, the cell destroys itself. And this sounds kind of silly a little bit, but what this is actually, this is a strong safety mechanism. Because you have like 30 trillion cells, and any one of them, could have the capacity, well, the nucleated ones, could have the capacity to become a cancer cell and cause a lot of harm. But if a cell has problems, so if there's DNA damage, for example, what happens is that they may not be receiving those survival signals properly, and so it's a mechanism whereby they'll destroy themselves. So they have to constantly receive a survival signal and respond to it. We see signals for growth and division, shown in red here. So a cell isn't going to divide until you tell it to divide. Differentiation. So we have all of these different cells, and keep in mind that the genes in those cells are the same. But some cells will be skin cells, and other cells may become part of your, I don't know, optic nerve. Okay? Others may be muscle cells. They have the same genes, but they're obviously doing different things, and that's caused by what we call differentiation. So they respond to a combination of signals, and they differentiate to become a specific type of cell. And we don't really look at that a lot in this course, but you look at that a lot in Dr. Chow's developmental biology course, which I think is Biology 308. So that might be something you'd be interested in later. And then the final one are death signals. So you can give a cell a specific, you can specifically tell a cell to die. And this is one of the things that our immune system does. It scans and looks for cells that are, say, infected with viral particles. It interacts with them and says, you have to destroy yourself, okay, for example. And so these are all important signaling processes. So this is a list. This was written up by the previous instructor of this course, and it's a nice synopsis, just the basic principles here. What are we looking at? And so, of course, a cell can only respond if it has a receptor. And so one of the things that may happen when a cell differentiates into a new type of cell, it may lose that one type of receptor. So it now no longer responds to a certain signal. Okay? And so that might be part of its differentiation, an integral part of it. And so if no receptor, no signaling. There's several types of signaling or mechanisms. The endocrine, this is signaling through hormones. Hormones are produced at generally higher concentrations than other types of signaling molecules, and they spread throughout the body, all right? And so those are hormonal signals. Paracrine, these are para means beside. These are localized signaling events or local mediators. And so these are generally at the concentration is overall is small, and so it sort of stays within an area. And if you move farther out, the signal doesn't spread that far. So that's paracrine. Autocrine, all right? So for autocrine, cells can actually produce signals and then respond to them themselves. So they release factors and then in turn, they respond back to themselves. Neuronal, so this is what we looked at in topic 5. Sorry, in your notes, I think it says topic 6. Synaptic signaling used to be topic 6, and I just missed that typo. And so topic 5, we've already looked at that, so we won't talk about that again. Contact dependent, the ligand is present on the surface of one cell attached to one cell, and the receptor is attached to another cell. And so the cells actually have to directly contact each other. We often see this in developmental pathways because you may have heard of terms of patterning, where tissues are actually made of a pattern of cells. And the way that works is the cells are actually directly telling each other, like, if I'm a type of cell type A, I need to be surrounded by type B cells. So everything I touch becomes a type B cell. Okay? And then around those B cells, you'll have additional A cells. And so this is how we can set up really complex organisms through patterning by this direct contact-dependent signaling. And direct, what we have is actually an open channel between the two cells that are signaling to each other. So they're directly sending a signal from the cytosol or cytoplasm of one cell into the cytoplasm of another cell. Questions so far? this is a nice picture depiction of this. And so at the top we can see the this endocrine signaling, so this hormonal signaling, where we release a whole bunch of signal into the bloodstream. In this slide, here's the autocrine signaling, so the cell is signaling to itself. The rest, it's hard to tell. These could be paracrine or contact dependent. It's a little bit hard to tell. If it was contact dependent, this ligand here would bind to a receptor directly on the surface, for example. If that was contact dependent, these might be paracrine. It's a little bit harder to tell in this particular one. But the important thing is that it's a localized, for the other types of signaling, it's a localized signaling pattern. So I'm going to start with the one on the right for intracellular receptors. So things like hormones are small hydrophobic molecules. So a small hydrophobic molecule, what does the membrane do for that? Does it prevent it from moving through? No. So these can move across, right, passively, okay, through simple diffusion. So they move all the way through a membrane. What's the problem, though? You have a small hydrophobic molecule, and you want to move it around in the blood. Can it move around in the blood? That's the problem. And so these hormonal signalings have to be moved by carrier proteins. And we have a whole host of different types of carrier proteins in our blood, things like albumin, and these pick up and carry hydrophobic molecules around the body. So they carry a hormone to the surface of the target cell. The concentration of that hormone is higher outside of the cell than in, and through simple diffusion it moves into the cell. Once it's inside the cell, it has the same problem, though, because the cytosol is a polar environment, and so it binds to a molecule inside the cell. And when it binds to that molecule inside the cell, we call that a receptor. And that receptor, when it binds to the hormone, is now active, and that receptor will mediate the effects in the cell, which very often we can see here. What happens is that receptor, when it's bound to hormone, migrates into the nucleus. The receptor does not move into the nucleus in the absence of the hormone. We'll look at those a little bit, but most of what we're going to focus on are what we see in the left-hand side, which are cell surface receptors. So these utilize hydrophilic or polar ligand molecules or signal molecules. So these move freely around in the blood or aqueous compartments. However, they can't get into the cell. So they are detected outside of the cell. So these receptors have to have some transmembrane component. They cross the membrane and they have an extracellular side, and that receives the signal. The signal is then transmitted into the cell, okay, but the signal itself doesn't move into the cell. So let's take a look at what that looks like. Here is our hydrophobic signal, and it's binding. This is cortisol. It moves through the membrane and binds to its receptor, and the receptor ligand complex migrates to the nucleus, and it activates target genes. Here we can see what happens with the cell surface receptor. So what happens here is that it sends the signal into the cytosol, but the signal itself doesn't move in. And one of the things it will sometimes do is sometimes it will utilize a secondary signaling molecule, such as cyclic AMP or calcium or cyclic GMP. So these are called second messengers or secondary signals. So it receives a small signal outside of the cell, and then it causes production of a small molecule, soluble molecule inside the cell, and that small molecule inside the cell will initiate changes in the cell's behavior. So those can be examples of second messengers, but we can also elicit, for example, post-translational modification of proteins. The receptors can do that. And so that can also result in these signaling pathways. Questions so far? So it's important to note here that in the classic system, the cell signal is received outside of the cell, but the signal does not itself move into the cell. So we're not transporting those little orange hexagons into the cytosol and having them do their thing. They're binding the receptor, and the receptor mediates the change. So there's 4 types of cell surface receptors. All right. The first one are ion channel coupled receptors. All right. And so what happens here is that we bind to a receptor, or sort of to a channel, and cause it to open or close. Similar to what we see with the voltage gated, but in this case, we're actually binding to a signal or ligand. And so we can imagine this happens super quickly. So you bind the channel, you open it, and we see immediate change inside the cell, an alteration in the concentrations of ions. And so that's one type of cell surface receptor. The next are called G-protein coupled receptors, and we're going to spend, I guess, next Tuesday's lecture talking about these. So really important class of receptors. And these are basically, the function of them is the same as a monomeric GTPase, but in this case, the GTPase is not a monomer, but this heterotrimeric protein. So we'll see how that works in the next class. But the idea, the way it works, the basis behind it is the same as for the monomeric GTPases. It's just a little bit more complex. There's a huge number of these. This is the largest single group of receptors, and they're really important for a lot of cellular processes such as growth, for example, and so they're heavily implicated in diseases. But they're also important as sensory receptors. So things like vision and smell, taste, these are mediated by G-protein coupled receptors. These two are like kind of the same, but not really. What we're looking at here are receptor tyrosine kinases, or receptors associated with an enzyme. So here what happens is that if we look at the top left, the enzyme coupled, what happens is when we activate the receptor itself is a protein kinase that will phosphorylate tyrosine residues. So we activate the receptor, and the receptor has enzymatic activity, okay, and it will act as a kinase. In the second example, when we activate the receptor, which is shown in the dark blue here, it activates an enzyme, such as a kinase. So the receptor itself doesn't have kinase activity, but it associates with a protein that has kinase activity. And so from the point of view of the effects in a pathway, those look the same. We end up phosphorylating target proteins, but the mechanism of action is just a little bit different between those. Yes?
Sorry, so could you please define what a kinase is?
Kinases, remember, add phosphoryl groups to proteins. Yeah. We'll spend quite a lot of time on those the week after reading break. Any other questions? Okay, so we'll leave it there for today. Don't forget Friday, no lecture. You have to watch your video from Kim. We'll see people on Tuesday.